Carbohydrate metabolism, pyruvate dehydrogenase complex. The oxidation of one molecule of glucose includes first the conversion of glucose to 2-pyruvate in the reactions of glycolysis. Second, it involves the conversion of the 2-pyruvate to 2-acetyl-CoA in the pyruvate dehydrogenase complex. And lastly, it involves the oxidation of the 2 molecules of acetyl-CoA in the process of the Krebs cycle. The Krebs cycle forms the reduced NADH and FADH2 compounds that then go through the electron transport chain. The proton gradient formed during the electron transport chain then powers ATP synthase and ATP production. In a prior presentation, we reviewed the key steps and regulation of glycolysis. Therefore, we will continue reviewing the next steps of the pyruvate dehydrogenase complex. While glycolysis occurs in the cytosol of the cell, the oxidative processes of the Krebs cycle occur in the mitochondria. It will be important to know the anatomy of the mitochondria, including the inner and outer membranes, the folds of the inner membrane known as the cristae, the inner matrix, and the intermembrane space. Both the Krebs cycle and the electron transport chain occur in the mitochondria, and it will be important to know where in the mitochondria that each of these processes occur. This diagram provides a visual summary of the processes of glycolysis occurring in the cytosol and the Krebs cycle shown in the mitochondrial matrix. And also you can see the protein complexes of the electron transport chain embedded in the intermembrane. Here again you can see glycolysis in the cytosol, the Krebs cycle in the mitochondrial matrix, and the electron transport chain in the inner membrane. We will now begin outlining the next step in the oxidation of glucose. After glycolysis, in the presence of oxygen, the 2-pyruvate formed are converted to acetyl-CoA in the pyruvate dehydrogenase complex. The enzyme catalyzing this reaction is pyruvate dehydrogenase. This is a simplified diagram of the pyruvate dehydrogenase complex, including the reactants and the products. You can see that the 3-carbon pyruvate loses a carbon through the release of CO2. And with the release of CO2, the pyruvate forms acetyl-CoA. In this reaction, oxidized NAD becomes reduced to NADH. You can also see that a molecule of coenzyme A is also used in the process.